Hello, welcome to the NIHR Dementia Researcher Midday Lecture Webinars. I'm Adam Smith, and today I'm delighted to welcome my friend Eric Dine. Hi, Eric.
How you doing?
Eric is a graduate student, graduate research student at Kent State University in Ohio in the US. Is it Go Buckeyes? Do I have to say Go?
Flashes.
I did have to Google that.
Yeah.
Eric and I met at the AAIC in LA almost a year ago, and sadly there was a free bar, so we didn't get to do much talking about research. But I have looked him up since, and we have spoken a few times since, and I think it's fascinating because it is some of the most amazing science, in fact verging on science fiction from my little knowledge of this, which is such amazing work that I had to ask him if he'd come and do a webinar for us today. So Eric is going to talk about his research into magnetothermal destruction of beta amyloid plaques. The talk will be about 20 minutes, and then we've got 10 minutes for questions at the end. As usual, I will put the questions to Eric and give him a chance to answer. You can post those at any time through the talk using the Q&A button at the bottom of your screen. We're also recording today's lecture as well, so don't worry if you have to drop out and can't stay for the whole thing. We will post this up to our website and onto YouTube probably sometime tomorrow. So thank you very much, everybody, for joining. Thank you, Eric. Over to you. You can share your screen now.
So hi, everybody. I'm Eric Dine. I'm a graduate student at Kent State University, and today we're going to talk about using hyperthermia via magnetic nanoparticles to disrupt beta amyloid plaques. As Adam said, it is quite a fascinating approach to targeting amyloidosis and Alzheimer's. However, I think this has a lot of very interesting implications, and we have a bright future for it. So let's get into it. So everybody is more or less aware about Alzheimer's disease and beta amyloid. It is a progressive neurological disorder. It's a leading cause of death in the United States of America. It's the sixth leading cause of death in the United States of America, and the first in the United Kingdom, which I found to be quite surprising. If we do not find care for this disease fairly soon, it is going to have a financial burden which will be most devastating in most countries. And so when we talk about Alzheimer's, usually you think about the cognitive decline. However, there are a lot of other symptoms that come with this disease, such as cortical atrophy, the extracellular amyloid beta, intracellular tau, and a few others. So what we're interested in is the amyloid hypothesis, which of course we have that amyloid precursor protein, and when it's cut by alpha secretase, we get a P3 fragment which can be cleared by the body and is not considered to be too much of a risk. Whereas when it is cut by beta secretase and then subsequently by gamma secretase, you lead off to this A beta fragment that is rather sticky and is prone to aggregation. So this image is from the National Institute of Aging in the United States of America, and we show here we have the APP protein. It gets cleaved by secretase, and as it comes off, it moves down from these monomeric species into these larger oligomeric species, forming protofibrils that then lead to larger fibrils that lead to these plaques. So these plaques are the target that we're interested in, since this is something that has been spoken about in Alzheimer's for quite a while. The idea that you have these fibrillary large plaques that interrupt the neuronal network. So there are two hypotheses for Alzheimer's disease, the amyloid hypothesis and the tau hypothesis. For the purpose of this talk, we're going to focus on the amyloid hypothesis. But however, there is still a great argument for the tau hypothesis, which we can definitely talk about further. I have a long dissertation on it that I think about the cause of AD, but for another time. So as you've already probably heard from a previous talk, microglia, the double-edged sword, microglia are responsible for clearing beta amyloid plaque from the brain, and that's what we are most interested in when we're speaking about microglia and Alzheimer's. However, these also have many other roles that are quite important, such as synaptic remodeling, axon trafficking, tracking, and also just the overall immune response to an insult within the brain. So the microglia have been found to be able to endocytose, or therefore phagocytose, beta amyloid fibrils relatively well in vivo. However, what we're interested in is, well, once the microglia uptake these cells, and they're supposed to undergo the clearance mechanisms through hydrolytic clearance, why are they not being cleared? We're not exactly sure what the purpose of that is or what the reason behind that is. However, we started to think that maybe it is simply just due to the size. There is research out of Cornell in the Maxfield lab that has shown that yes, they do take them up, but then after about 2 weeks, the beta amyloid plaques within the microglia will eventually promote death of the microglia and then be released. So we came up with a manner of how can we actually target beta amyloid, we realized that we needed to find a way to target beta amyloid, as we have a hypothesis that this is going to be a leading cause into the etiology of Alzheimer's disease. So what I'm showing here is, this is a microglial cell without any beta amyloid. This is taken using scanning electron microscopy. So an otherwise healthy looking cell. But once we add amyloid beta, we see this. We see that the cell morphology undergoes a drastic change and they look rather unhealthy and dystrophic. And during this time, when microglia are exposed to beta amyloid, we see an increase in pro-inflammatory cytokines such as TNF-alpha, IL-1 beta, IL-6, and several others. We see an increase in reactive oxygen species, ROS, and we also see an increase in nitric oxide. So knowing this, if we cleared this, would that affect the pro-inflammatory response, reactive oxygen species, or nitric oxide generation? And then this is just an image of our cells using a confocal microscope showing the co-localization of the beta amyloid over time. So there's been a lot of research into targeting beta amyloid using some type of immunotherapy. And as of right now in the United States, the Food and Drug Administration has approved only 4 drugs, all of which are cholinesterase inhibitors. And the issue with it, and that's fantastic. However, none of these are disease modifying, meaning that after once you take these drugs, it doesn't necessarily change the course of the disease. The disease will continue to progress over time. And so there's been a number of great trials, including aducanumab, which is again coming back to, I think for the 3rd attempt there, Biogen and Eisai are bringing back aducanumab to clinical trials, and we're hoping for the best success. But as of right now, all of these drug trials have failed. So we started to think about another approach to potentially clearing beta amyloid from the brain. And this was stemming from the idea of hyperthermia. So what is hyperthermia? Hyperthermia is effectively inducing a change in the temperature in some type of localized area. So it could be the brain, it could be within the liver, it could be targeting a solid mass, a tumor, not exactly sure. So not sure, but we don't know where exactly it will be. Has a lot of possibilities. So there was a study in 2015 by Loy Netchan et al. At MIT where they showed that they can induce magnetic nanoparticle AMF experiments, meaning magnetic nanoparticles, and then alternating magnetic field generation. I'll talk about what that means in a moment. And they were able to show that it is possible to target beta amyloid and using nanoparticles and producing heat to change the overall structure of the beta amyloid. And that, coupled with our laboratory success with Staph A biofilm susceptibility to antibiotics when exposed to nanoparticles, we thought this would be a novel target that needed further research. So what is the whole idea behind nanoparticles, magnets, heat, and beta amyloid? So what we do is when we insert the nanoparticle into the body, or into wherever the location is, we apply an alternating magnetic field. And that alternating magnetic field will cause Neal and Brownian relaxation of the nanoparticle, which will cause motion. And that motion will then create heat. The closest to the nanoparticle surface, where we see this red line, is going to be the best effect from heat. It's going to be quite high, but then we also have to consider the ambient heat, which is going to be this yellow line, which this will be the heat that is going to be most likely exerted on the surrounding tissues and cells. So we want to make sure that microglia, astrocytes, neurons, the brain tissue itself, everything is relatively safe. And we actually have a way to quantify that using a calculation called CEM43. And I'll talk about that in a little bit. And this is actually our results from our different concentrations of nanoparticles. When we change the concentration of nanoparticles, it changes in a dose-dependent manner. So the higher the concentration or dose of nanoparticles, the greater the change in temperature that we see. So a little bit about what we did. So and also a little bit about nanoparticles. We use iron oxide nanoparticles that are found to be biocompatible, and we can easily conjugate these nanoparticles with some type of ligand or pharmacological agent, so we can find a great antibody to help target beta amyloid. And doing some pretty basic chemistry, we can actually add them to the surface of the nanoparticle to induce a greater effect of these nanoparticles on the target. And part of our research is actually working right now. We're conjugating different drugs to our nanoparticles, but I don't have enough time to really go into that. So if you have any questions, I'm happy to share more. But here's our overall schematic. We take the nanoparticles, we add them to the culture of our microglia. Then we have our electric magnetic field generator. We add the samples into these coils, under which they will undergo the magnetic field, which then induces this alternating motion, and then that's going to create localized heat. And then afterwards, these large amyloid beta plaques then come out, and they're much smaller. And then taking out the nanoparticles using a very strong magnet or some other separation method, we're then able to treat the microglia, the new amyloid beta with the microglia, to understand the effect on size on microglia. And then we've done this in multiple different ways. We've stimulated the microglia and then added the amyloid beta, just add the amyloid beta. I've done a whole bunch of experiments to understand the behavior of the newly fragmented beta amyloid. And we even done some experiments where the microglia plus nanoparticles were concurrently exposed to the magnetic field. So we've done a lot of very fascinating research on this. So just a quick reason why we're doing this. As I mentioned before, the drugs that we have come up with so far for Alzheimer's disease or trying to treat Alzheimer's disease have unfortunately not worked. So we're trying to approach an alternative method to overcome the limitations seen with the clinical trial drugs. And so again, this is a quick schematic of what we do. So just then the overall goal for this project that I'm going to talk about today was to develop and validate a use of mild hypothermia via magnetic nanoparticles for improved clearance of beta amyloid. So here we have a plaque that we had synthesized. And then after 15 minutes and 1 nanogram per milliliter of magnetic nanoparticle, this was the end result. And you can see relatively well, you can still see the nanoparticles in this image. So we were quite excited after our first trials to see such a robust change. So we had a couple goals that I would like to go over and just to show what we did. We understood that the beta amyloid over time will self-nucleate and then grow into larger species. We had attempted to work with some oligomers, however, we found them to be incredibly unstable, and we ended up, before we could even get to treatment without adding some type of blocking group, that our oligomers were already turning into small fibrils. So throughout this talk, we're just going to focus on the effect of fibrils rather than oligomers. However, there's a lot of fascinating research on beta amyloid oligomers. So quite a little bit larger here, we see the same schematic as before. And so what we were interested in is, could we first synthesize the beta amyloid, and then could we also size it? And so once we synthesized the beta amyloid successfully and did a bunch of imaging, which I didn't include because there's just so much to show, we ended up looking at what is the effect now on the beta amyloid. So we did dynamic light scattering, we did scanning transmission electron microscopy, as well as some confocal, and we started to look at the effect on the beta amyloid. So this here is actually of the large untreated beta amyloid, just to show the effect on the cells. As this is the same one shown before, just without the IBA1. So you can see these are relatively large plaques that co-localize with the nucleus of the microglia. And then when we ran the size, we looked at a 16-hour incubation, meaning this is going to be our largest fragment. So when we do the normal preparation of the beta amyloid, which is 16 hours at 30C, we come out with somewhere around 3 micron average size, which the literature supports. And the longer we go, the larger they get. But then we also looked at the effect of smaller. And so these were smaller plaques that we also worked with. And I got some data on that too, which I can talk about if necessary, but we're going to focus mainly on the large plaques today. And then also just looking at various species. Of course, this one was treated with nanoparticles. So we had to determine a safe concentration for for applying heat. Too much heat could become cytotoxic and damage the cells. Too little won't have the proper effect. So as shown before, we did different concentrations of nanoparticles and then calculated the change. Using this, we plugged it into this equation, or more simplified equation here, where we're looking at the change in temperature, and then we have constant. When the temperature is less than 43, this is 0.25, and when it's greater than 43 degrees Celsius, it's 0.5. And then we calculate this by the change in time, which is half-minute intervals. And then calculating that, we can identify this number, this thermal dose known as CM43. And the literature has shown that about a 10-minute CM43 is going to potentially become cytotoxic to brain tissue. So we found that all 4 of our treatments were sub-threshold, which is fantastic. So we could use a pretty robust concentration. However, after more experiments, we determined that the initial concentrations of test are going to be 0.5 milligrams per milliliter as well as 0.1. And this is what the core looks like on the actual machine that we use. So next, we needed to understand, can we co-localize the nanoparticles around the beta amyloid plaques? And we did this using electron microscopy again. And then we showed that yes, when we just incubate the plaques themselves into the culture, they indeed do co-localize with the plaques, the nanoparticles co-localize. However, we also could use a targeting agent, which is something that we're looking into, and we've so far had some pretty decent success increasing our targeting towards beta amyloid. However, without any targeting agent, it seems to localize quite nicely. So then again, I included the image, what happens when we have the plaques before we apply the AMF, and then afterwards we see a much smaller. I do have to apologize. I had to kind of sort of limit the number of the amazing photos that we were able to include due to we're still publishing this work because of the coronavirus. We had a 3-month hiatus, so we're just about to submit the paper, and we don't want to give away all the figures yet, but it'll be out soon. So after doing that, we realized, okay, so we can successfully break up the beta amyloid. However, I was curious, what is the effect on the microglial cells? So a lot of studies have looked at using confocal imaging and other types of quantification. However, I thought, well, I'm not getting the detail that I'd like to see. I would like to really get an idea of how the microglia are reacting. So what we did is we used scanning electron microscopy rather than traditional microscopy. And I looked at the effect of untreated microglia, compared it to microglia plus alternating magnetic field plus nanoparticles. And this is our machine that I was just on a few days ago for a nice 10-hour day. And what we see is we see these large projections as normal, a pretty healthy looking cells, smaller projections, primary, secondary, tertiary, nods off of them, pretty good, smooth surface. And then we got up close and personal with those that were exposed to the alternating magnetic field without nanoparticles, and basically looking at all the images. It took about 50 images per trial, and then we used an algorithm to calculate the distances and the cell body size. There isn't too much of a change actually between these cells. However, when we have the nanoparticles have been shown to activate microglia. So something that we weren't too surprised about was we started to see these major projections decrease. And then ultimately we end up seeing these two bipolar, we see a bipolar microglial cell here. And then we see a number of the smaller projections disappear. So next, we wanted to quantify the uptake and clearance of beta amyloid facilitated by mild hypothermia. Therefore, the idea now is that if we have smaller plaques, that we're going to be able to, the cells will be able to uptake them with a greater efficiency and then clear them. So how we did that is we took confocal images, we took Z-stacks, and then took the Z-stacks, and then we did digital colorization and added in the area per space, and we were able to then use a computer macros code that will calculate the amount of the cell that is positive for beta amyloid and then the amount that's negative. We also did a couple other programs that were able to help us with this, looking at the entire image, calculating intensities, calculating number of cells, as well as using flow cytometry. So what we showed is that when we added the 1 micromole beta amyloid plaques plus 1 nanogram per milliliter nanoparticle for 50 minutes at AMF, over time we see a decrease in signal. And this is just 3 of our results. Like we have a couple more of them analyzing, but I see this. We saw the same result each time, as well. When we look at the percent of THT positive, so thioflavin T is a stain for beta amyloid. When we look at the treatments for the 0.5 and 1, we do see an increase in F from 2 to 24 hours, which is most likely the cell actually taking up the beta amyloid fragments, regardless of size. However, what is different from the control is the 0.5 and 1 milligram per milliliter nanoparticles both show a decrease in signal over time, whereas the controls show an increase. By 72 hours, almost every cell was positive for beta amyloid, and this was determined both using confocal as well as flow. And then this is some of the flow data. We have a lot of flow cytometry data. where we compared controls. So this is just beta amyloid. This is just the beta amyloid without nanoparticles over 72 hours, 24, 20, 48, 72, compared to a negative control. And then we did the same test with more controls this time, because I wanted to have a comparison per time group to make sure that we didn't have some type of variation due to light exposure or something. And we showed that at 24, 48, 72 hours, the signal decreases in such a manner. So that was positive, but then another positive outcome we found was actually that the pro-inflammatory signaling from exposure to the nanoparticles and the smaller fragments actually attenuated gene expression. And these are just some of the genes that we looked at. TNF alpha, comparing the control to the 1 micromole of A beta, we saw a significant increase. However, over time, we saw that decrease. I mean, there still is a significant increase comparing the 1 micromole beta amyloid plus the 0.5 nanograms. However, there is no significant difference between the control and then the 1 micromole A beta plus the 1 milligram per milliliter nanoparticle, which is positive. And then again, we saw similar results with IL-6, a robust increase and then a decrease. And then we also saw a robust increase in IL-1 beta and then a decrease. So we're pretty happy about the outcome of this study so far. So what is the next steps for this project? Well, we're moving this into an in vivo model. We're going to be using the 5 transgenic mouse model. We've done some preliminary experiments looking at the nanoparticle distribution as well as targeting the brain to do some safety efficacy tests. So we're going to start with that. We're going to induce the nanoparticle concentration to the brain at first without targeting, but then later with targeting, as well as some other fascinating approaches in order to ensure the proper delivery of the nanoparticles to the area of interest in the mouse. And then we'll do some similar experiments, but even further, we'll go into some immunohistochemistry, do some neuropathology. It'll be quite exciting. As I mentioned, we're also looking at potentially conjugating an antibody of beta amyloid to the nanoparticle, doing some chemistry. And we have a team of chemists who are going to help us with this too. But so far we've had some pretty good success with this. And then another thing we're interested in is drug repurposing. We've been able to look into some other drugs that have been so far tested for Alzheimer's that haven't had much success, or even some other drugs that haven't ever been approved to Alzheimer's. And we're trying to repurpose them to be used in this model. Since we can have a direct delivery of these nanoparticles to the target, it offers us a pretty interesting approach to looking into how drugs interact when they're not affected by barriers or other types of issues that come along with treating or getting drugs to the brain. So where can we go with nanoparticles? The future is vast. They can be made out of many different materials. We can stimulate them using various manners. They're now using these for contrast agents, drug delivery. And I think I said kind of the same thing with the brain, but yes, they're looking now into using nanoparticles for glioblastoma. They're using it for Parkinson's disease. They've found some success with delivering drugs for oligodendrocytes and multiple sclerosis and some leukodystrophies. And then the question that I get constantly is, well, what about tau protein? We're focused on the amyloid hypothesis. What about the tau hypothesis? And luckily there's already groups that are looking at targeting tau or using nanoparticles to negate the effects of tau protein. So the field is growing. It's relatively small at the moment, but it is a very exciting field. And the cancer therapeutics that come with nanoparticles and mild hypothermia is just fantastic. And what am I doing after I graduate? I don't really know. I haven't gone that far. I'm very interested in a lot of different things. If anybody sees this and thinks, wow, I think he has potential, please do give me an email. And of course, I have to plug for my poster at AAIC. If you're interested in hearing me talk about this and asking more questions, please do attend my poster session on Monday, July 27th. I'm poster 44183. It'll be very similar to this PowerPoint, I think, since it's only a month away. And I want to thank my very large group. Team science is fantastic. I have to thank my PI, Dr. Kim, everybody in our lab, my dream team undergraduates. They're fantastic. I really appreciate undergraduates. These guys are they're the best. So I appreciate them. I have to thank Dr. Huang. He's our chemistry component, and Rong Wei Song, as well as Jun Feng. And then our new collaboration with Schmidt Lab, who does the most amazing DNA nanostructure, like DNA origami work. It's fantastic. And then my collaborators at Case Western Reserve University in Cleveland, Ohio, University of Georgia in Athens, Georgia, University of Pennsylvania, University of Pittsburgh, both in Pennsylvania, Stanford in California, and University of Nevada in Las Vegas and Las Vegas. Do you have any questions?
That was fantastic. And well done on the graphics as well. That must have taken you ages to draw all that out. We do already have some questions, although before I go to those, I'm going to come back to you. So I suppose the next stage in this mouse work will allow you to see just how safe the potential delivery and practicalities of this is.
Yes,
human. I mean, that's where I'm going. This is with the human element of this.
Yes, and so I should mention too that we use human microglia. So that's something that's unique to our research. Is my collaborator at Case Western Reserve, David Alvarez Carbonell from the Kern Lab, has really done me a monstrous favor by providing an immortalized human cell line, so that when we do the microglia work, we're not using a mouse model. This is actually human cells, and we use human amyloid beta. So we try to keep our research as close as possible to what is physiologically relevant. And so I have to definitely appreciate that. And yes, when we move, but of course we can't do this in humans, so we have to move back to the mouse model. And I have very high expectations for the mouse work. What we have done so far is just simply done a couple experiments to look at the feasibility of doing direct delivery of nanoparticles to the brain. We've taken some brain sections, and I wish I could include them. I actually even have them on this computer. And surprisingly, the co-localization is not that bad. When we did the surgery, I was afraid over time they were going to spread throughout the entire brain and we're going to lose the effect. We actually saw that over 72 hours, it still remained relatively close to the target. So we will be moving to that next. The big question, of course, is going to be, and we've done this also with the biofilms, but it's going to be what happens when we actually put the brain inside the alternating magnetic field and then we apply the current.
Yeah, that's the bit that kind of, I mean, you've got to develop I mean, just out of interest, just a piece of kit. Obviously you gave us a picture of the machine there. Does this machine exist on that size, that scale, or are you gonna have to create a new coil?
Well, we have that. So that coil is enough to fit in the animals, the mouse model. It will fit quite nicely. For a human, they have not, I don't believe that is being currently conducted in the US, but I do believe that in Europe, they are actually using mild hypothermia in various manners for treatment of cancer, particularly glioblastoma, as I mentioned. It's been absolutely fantastic. And so, yes, we have multiple coils. We have 3 machines, with two of which that are incredibly powerful. Well, I should say we made one small tabletop one that I wish I had a picture of, and I wish I could actually bring it in because it's just sitting on the table. We made a small tabletop one to see if it was possible to have a portable, and it wasn't strong enough, but it was enough to heat a thermometer very fast. Or if you took a metal pot and then put some water in it, it boiled almost immediately with once we added the nanoparticles. And so, well, also the heat itself from the metal, is incredible. And then we have the one that we use, which was from MCI Automation in the US. And then we have another custom built one it's 4 times as strong. And that one is very, very powerful. And that one, we had some data on that. We originally started on that machine. It was so quick to reach the temperature that we needed. So the one we have is a lower power. I forget the characteristics behind it, but I think we're around 150 I think we're doing 150 kilohertz per minute, I think.
So the scaling up's not an issue. I mean, it's the same thing. It's just a case of making this bigger and putting some cushions around it.
Yeah, pretty much. Yes, we actually shoot water through the coil. That's how we keep the coil from getting really hot. This system has a water cooler that is running while the machine's on. Otherwise, that coil will get really hot, and we have it insulated so that we can safely put tissue in there without it getting burned from the coil.
Some nice grant there to build this new kit. I mean, this is what I find so fascinating about this. This really does feel like cutting edge, and I've not seen that this is happening. I could be wrong, but in many other places as well. It seems like you are kind of leading the way on this. Okay, let's go to the questions. Enough of my chat. Chloe Tulip asked a question quite early on in your talk. How do you get the plaques in vitro? What's the harvesting process?
Oh, so what we do is we actually buy from Bachem. We do buy commercially available beta amyloid plaques, and then our protocol we use. So we that's another great question is how we prepare it because there's so much variation when it comes to preparing the beta amyloid. But ours, we have a solution which I could give the recipe to. It's quite long, but initially to be simple, we take the plaque and then we're going to add HFIP, and then that's going to help break the bonds and then try to monomerize it. And then once we have that, we let it dissolve into a film in the fume hood overnight in the dark. And then once we are ready to move, we add a buffer. We have our own buffer that Jun Fong had helped us create that is quite special, but it's acetonitrile, a special phosphate buffer, as well as, I'm forgetting the last compound. And once we add that mix, about 200, we make a 20 micromole solution, and then we put it into a 30°C stationary incubator. And then after 16 hours, we take it out, and that's what we call our large plaques. We found that 16 hours, we get consistent results with the size. When we tried 37, it just went quicker. The kinetics of the aggregation were a little bit quicker. And then if we did agitation, it also induced it induced the size quicker, but it also had a bit more variation among the size. So when we did the stationary 30°C, that was best because then every time we have almost a uniform distribution of beta amyloid. So we use the Bachem human amyloid. So we're not doing our own protein isolation, but it's something that we may have to do in the future with the animal models. Since we're going to be going in vivo, we have a protocol for the protein isolation, which I can share.
Thank you, Eric. Thanks, Chloe, for the question. I do hope that answered that. Remember, if anybody's got any questions, you can use the Q&A button at the bottom of your screen to ask those. We've got a question from Fiona. You used chat, Fiona. It's confused me for a second. Fiona McLean says, really interesting work. I was wondering if you have considered how amyloid plaques may respond differently depending on if they are located primarily near neurons versus microvessels in the brain?
Fantastic question. So it's something that we have to consider. That is definitely a consideration for the study is the localization among where we're putting these nanoparticles. Because obviously if the beta amyloid is within the cell or in very close proximity to a neuron or an astrocyte, it's very likely that the nearby glial cell will experience the effects of mild hypothermia. Since that change in temperature we showed is just the ambient temperature, that's the temperature of the system changes about 6 C at our highest concentration. When we're looking directly next to the nanoparticle, which would be co-localized to the beta amyloid, it's much hotter, and it would most likely damage cells. So that is an area of interest. And then, of course, with the vasculature, with cerebral amyloid amyopathy, we're quite interested in that as well, since we would easily be able to induce the nanoparticles within the bloodstream. It would then become a question of targeting the nanoparticles to the amyloid and then also the effect on the vasculature, because obviously we don't want to burn any brain vasculature. That could become catastrophic. So that is something that we definitely need to think about more so, and a very valid concern. And hopefully this research, if it continues and we show promise, that will become an area of interest that somebody will look into, because I don't actually have the answer, but in case of neurons, we do need to consider the safety of the cells and the surrounding, that it will be quite hot, and we don't want to promote more death because there's a possibility that if we end up destroying a bunch of neurons, that may exacerbate tau pathology.
Yeah, so also as well, then looking at the different types of dementia, I suppose, and where in the brain the amyloid is as well. Yeah. Mm-hmm. It's fantastic. Thank you for your question, Fiona. Please do, hopefully that answered that, but please do ask more if you have more questions. We have another one from Daman Kerr, who asks, what's the circulation time stroke half-life of the nanoparticles?
I'm not actually sure. Well, right now, since we're doing in vitro, it is when it's simply however long they're in the sample. So we isolate them using a magnet when we're finished. If we're doing dynamic light scattering, we don't want to have interference from the nanoparticles. When it comes to the brain, that is something that has not yet been addressed. That is on our that is part of our protocol that we need to address. So that is something that we haven't gotten to yet, but that is a fantastic point and something that we will be looking into for that exact reason. Because so far we've shown that after 72 hours, they continue to localize within the site of injection within the brain. However, it is very likely that it will be cleared through the brain's lymphatic system or throughout one of the ventricles or the CSF, very likely. And so that's a great question. And there has been some research into brain treatment of nanoparticles, but the majority of it has been in solid tumor. So in which case they're being uptaken by the tumor, and then after which time they haven't looked too much into it. But I believe there might be one study, but I have to look into it. I'm not 100% sure about that answer.
That's all right. Thank you very much, and thank you for the question, Taman. Actually, that's a good question, Eric. You mentioned before about the potential of using this delivery mechanism for some of those drugs that perhaps have found that their issue was more with the mechanism of delivery rather than the drug themselves. Have you had much interest in that? Have you put feelers out? Have you had much interest?
You know, I'm actually writing an email right now to Biogen and hope that I can potentially get access to one of their clinical trials. And I've already tried with one pharmaceutical company, and they said, well, unfortunately, we can't provide it right now, but it's a fascinating idea that you have. And I would love to, and I have some ideas about how to do so. We have a protocol about how we can kind of, it's kind of long, but we should be able to hypothetically cross-link a carrier group to the nanoparticle surface, and then we can bind on an antibody or some type of drug target. And I thought, in my eyes, I see this as a fantastic opportunity because we're now bypassing a lot of the issues that are seen with the drug delivery, such as getting through the blood-brain barrier, permealizing it, finding its target. And so we're doing stereotaxic surgery, so we're able to go directly into the brain and then put it right where we want it to be. And we can use other imaging as well to localize an area of high amyloid and then add even greater specificity if necessary. So I'd like to look into some of the possibilities. Aducanumab, BAN2410, a couple of the other ones.
There's quite a few. I mean, I suppose if you only have to look back over the last 30 years, I mean, there's plenty to choose from there. Yeah. Haven't seen the success that they'd hoped for. Well, honestly, Eric, that was fantastic. Thank you ever so much for agreeing to share. I'm going to put my notes here on the screen so I remember what I'm supposed to say now. Thank you, Eric. That really was fantastic. Thank you for agreeing to share. It kind of really does feel like it's verging on the sci-fi piece there. Maybe you should be approaching somebody like Elon Musk for your next round of funding. I get a feeling that this is something that he might be very interested in. It use a tricorder then to get rid of them, to put them in there. Eric is new to Twitter, but you can find him on there. He's on there now at last. His Twitter name is @Glia, Glia Alzheimer's. And I know, as you said at the end there, you're looking for fellowship opportunities. Mm-hmm. So please do keep him in mind, drop him a line, and do have a look at his poster during the AIC. Our next webinar takes place at 1 p.m. on Friday, the 10th of July, when we have Mohammed Abdulli discussing a bridge between cognition and olfaction, which is I just got distracted there. We had another somebody's asked another question. I'm going to finish my bit first. Recording from today will be and details of future webinars are all on the registration page of our website, which is DementiaResearcher/webinars. You're going to have to be quicker in future, Johnny. Amazing talk. 2 questions. Thinking about implementation in humans, how are the nanoparticles cleared once they dissolve the amyloid beta? There is another question, but I'll let you address that one first. I guess, do you know yet?
Well, our study, we haven't been able to validate that, but from what I read in the literature, the belief is that it will be cleared just as anything else within the brain. It will be removed via the cerebral spinal fluid, most likely over time. We have some questions about that because there's, of course, the concern of nanoparticle pollution from the environment that they've seen post-mortem. Some brains have very high, different metals in the brain. And so the question is, well, why are they not removed? Why are they aggregating and can they be removed? But hypothetically, being their relatively small size, they will be removed. It's just a matter of how much will be moved over what time. So we have to, that's still a question I think that needs to be
looked at. Yeah. I mean, you get a sense that this is still very early days with this, isn't it? I mean, there are so many other questions to ask off the back of this to prove the safety, particularly in its application. The second part to his question is, given the increases in temperature is an innate trigger for immune response, how could hyperreactability be prevented? Example, considering that microglia is already hyperreactive in AD.
That's a great question. So that was actually one of the most interesting finds to the study was the change in inflammation. So research has shown that nanoparticles do induce inflammation. It's not exactly robust, but there is an increase in inflammatory signaling when exposed to microglia. However, when it came to hypo or hypothermia, it's been really interesting because each one of them have their own role in Alzheimer's disease. And we have found, and others have actually seen, that heat shock proteins may be actually helping in this manner. There was a study in 2015, I forget the name, first author, but they looked at heat shock protein exposure and the rate of clearance by microglia. And they showed that heat shock protein exposure actually induced clearance of beta amyloid or promoted it. So we are thinking that it's possible that the nanoparticles are activating heat shock proteins. And that's a very big part of my research that we haven't gotten a lot done. So I was going to kind of keep that on the DL, but why not? So yes, we think that the nanoparticles are potentially activating heat shock proteins, which then may be assisting with the immune response to beta amyloid, that it's actually being more of a help than a detriment. But it is also true that heat does activate microglia, as does beta amyloid, as does the nanoparticles. So there are a lot of variables at play when it comes to this experiment. However, with that in mind, we took the samples as is. We ran gene expression analysis multiple times, and we saw the same pattern. So we believe that compared to just beta amyloid alone, the administration of the nanoparticles plus the AMF, the mild hypothermia, is enough to attenuate the inflammatory response. And hypothetically, if we can attenuate the inflammatory response, that's going to promote, that could potentially downplay tau, but I'm not sure about that. That's just hypothetical at this point, that maybe if we can slow down, the amyloid pathology, that might halt tau, but there's not enough evidence to make that statement yet.
Thank you, Eric. And thank you for the question. Where was I? Oh yeah, well, thanks again, Eric. Honestly, it's fascinating. We'll be recording today. If you're already watching this on YouTube, too late. We've already had it live. But you can register for future webinars as well at Dementia Researcher /webinars. Also, to let you know, as I mentioned before we started recording today, we're running a small competition throughout July 2020, which is to win a pair of Beats headphones. To win, you just have to have registered and attended one of our webinars in July. You have to leave a review of our podcast on iTunes, which you'll find there using searching for Dementia Researcher, and be registered on our website as well, which is dementiaresearcher.nihr.ac.uk. And we'll draw a winner at the end of July. Last of all, if you'd like to join us and present your own lecture webinar, we're always looking for people to come and talk about their work or career topics as well. Just drop us a line through our website using the Contact Us button on our website, or tweet us at dem_researcher. Thank you ever so much again, Eric. Fascinating stuff. And please do reach out to Eric if you through Twitter if you've got more questions today. Thank you very much. Bye.